So this is the electron transport chain. So, whoa, sorry, did not mean to do that. Electron transport chain, which I will, but not just me, but a lot of people also abbreviate it as ETC. So later on, if you see ETC, it means electron transport chain. And then the oxidative phosphorylation pathway, which a lot of people call OxPhos. Okay, so just in case I say OxPhos or ETC, you'll know what it is, or you'll know at least what I am referring to. Oh, where does it occur?
The lining of the inner mitochondrial matrix.
The lining of the inner
The
mitochondrial.
mitochondrial matrix into the inner that was space.
So, okay, the mitochondrial matrix.
Yes, right there in the membrane.
And the inner membrane.
Yes.
Anywhere else? Does any other place have any role whatsoever with it? So if we draw our little handy dandy Millie the mitochondria.
Is it also the outer mitochondrial membrane?
No. So now let's say, but yes, you're right. Here's the mitochondrial matrix. I don't have that much room. So part of it does occur there. Part of it does occur in the inner membrane. So, through here. And then there is one role here in this portion right there. Whoops. What is that called? That's the inner membrane space, so the IMS. I shouldn't say it's relatively minor. It is important. It happens there, but yes. But it does not have anything to do with the outer membrane. It doesn't have anything to do with the cytosol or anything like that. So if you're making a big, picture of the cell or whatever, and you're putting in different pathways and stuff, you'd put this one in there. This one also doesn't have a nice little shape, a nice little circle or anything like that. It just looks, it's different. It's its own thing. All right. What is the purpose of the electron transport chain? Whenever those it's to transport electrons. And also transport something else. What else does it transport? Pardon? Because it can't just transport electrons. Okay, so what else does it transport? Protons. Okay. And then what's the purpose of oxidative phosphorylation? To make ATP. So then these two pathways, if you want to call them that, are coupled together usually. There are times when they become uncoupled, and the proteins or enzymes or anything that uncouples them are called uncouplers. And so but normally they go hand in hand. That's why they're usually put together. Okay. So some books will tell you that there are 5 complexes of the ETC. That's really an asterisk, okay? Really, there are 4 multi-subunit complexes, and then we have cytochrome C. So that's why I just wanted to put that out there. Cytochrome C is a protein. Coenzyme Q is not a protein. Right. But yeah, so we have 4 multi-subunit. These are huge multi-subunit complexes. Their names are relatively, well, it depends. You can either think of them being really, really simple, or think of them as being very, very descriptive, because they actually have more than one name each complex. There's cytochrome C, which, as I mentioned before, it has a cytochrome in it, and it's a protein. And then there's coenzyme Q, which we've seen something like that just in the last chapter when we talked about the quinoids. This is similar to that. That's what Q is for. That makes what the quinol, quinone, and semiquinone. Those are the 3 states for coenzyme Q. And this is just a huge slew of redox reactions in the electron transport chain. Obviously, it's not for phosphorylation, it's phosphorylation. Ultimately, this is usually, I don't want to say it's not meant to be a trick question, but if I've asked it before, people have gotten stumbled. If I said, what is the ultimate recipient for the electrons, the electron transport chain, I've seen so many people say like ADP or ATP. That's wrong. That's phosphorylation. The ultimate recipient is oxygen becoming water. Okay. ATP is not made in the actual electron transport chain. It's in, some people call it complex 5, but it's not. It's really sent by the ATP synthase, which is a completely different complex. All of the NADH and FADH2 that we've been making in all these other processes, this is where ultimately you want them to go, and they're going to enter in different places along the electron transport chain. That's why you make more ATPs using NADH than you do for FADH2. It will make more sense once we actually see the entire process. But ultimately, FADH2 or NADH are going to transfer electrons through a whole series of different places, including coenzyme Q, until it ultimately reaches oxygen, and oxygen is transformed into water. Okay. And you need to know which steps pump protons. That's very important. But some of the steps will pump protons from one place to the other, and because of that, it creates a chemical gradient, electrochemical gradient, and that's what drives ATP production. Because remember, to make ATP, and to make lots of it, which we've seen, so many things have required the use of ATP, to make enough of it for you to live and to be viable, you have to be very efficient. So mitochondria are very, very efficient if you're healthy. If you have some type of cardiomyopathy, mitochondrial defect. One example I'll show you is in complex 3, typically a healthy person, it has to be able to transport electrons between 100,000 and 150,000 times per second. Okay, so I mean it's going fast. Okay, this is just, I personally think this picture is ugly, but it does explain a lot of this, a lot of what's going on here. I don't know, it just looks like something from the 60s or something like that. Maybe we need to jazz it up a little. So I'll give you a moment just to look over it on your own, and then I've asked a couple of questions on this. I'll point out, I do like one thing about the way that this one's set up, because it's something that people often get wrong, but I think that they show it nicely here. So look over all of it. We're going to be drawing the membrane as we go along. A lot of times I used to draw it on the board. It would take up a lot of space. I tried in the past to draw it here on my iPad, but I can't draw that tiny as we go along. I may try again. Okay, so first I want to point out some of the structural features. This right here is the outer membrane. It's not labeled, so therefore this is the inner membrane. The space between it's called the intermembrane space, or I like to abbreviate it, and other places also abbreviate as IMS. So this may be abbreviated as IM and IM. OM and IM, and this is IMS, just in case I start making those abbreviations whenever we start drawing things over and over and over again. We've already talked about how the inside, that holy of holies of the mitochondria, is called the mitochondrial matrix, which is where the Krebs cycle is occurring. Let's see, and then they're trying to show here the phospholipid bilayer of the inner membrane. Okay, and remember, so far we've been showing how even NADH that was made or synthesized in the cytosol, which would be out here, it can't pass through the membranes, but remember we use malate in order to, malate dehydrogenase in particular, in order to ultimately transfer them onto the inside. The electrons from NADH here gets transferred to make malate, which goes on down here, which goes back to oxaloacetate and gives its electrons to make the NADH. Okay, so I'm just going to walk you through this entire process. This is the oxidative phosphorylation, so we're just ignore it for the time being. If you notice, it looks like there are two different electron transport chains. This is the common mistake that gets made by students because they know the simple names. There's complex 1, complex 2, 3, and 4, and then there's, like I said, there's cytochrome C. Let's see, how can I state this? What happens is, if you notice, the way it shows here, it goes from 1 to 3 to cytochrome C to 4, and down here it goes from 2 to 3 to cytochrome C to 4. That's because it really matters which of these two possible chains that you go through, that whether you start off with the NADH or if you start off with the FADH2. If you start off with the NADH, you're going to start off at complex 1. The electrons, of course, it goes from NADH to NAD+, but the electrons get passed off to coenzyme Q. And to some, and I don't want you to worry so much about the numbers here. Some books will say it's 2 hydrogens, 2 protons that get pumped through. Some will say it's 4. Okay, there's some debate on the actual stoichiometry, but there are multiple hydrogens. Okay, that's why I want you to know there are multiple protons that get pumped across the membrane. Coenzyme Q, okay, then becomes reduced, and it goes. Notice it does not go from complex 1 to complex 2. It goes from complex 1 to complex 3 if you start with NADH. The electrons then get passed here. Once again, multiple protons get pumped across. Then the electrons get passed to cytochrome C, which when it's reduced, it goes over here to complex 4, which passes the electrons off. And it's complex 4 that actually will turn the oxygen into water. Okay, just like some books will show with this, that they don't like to use the half, because you probably learned in Gen Chem, we don't use those stoichiometries of fractions. So it may say 4 plus 1 goes to make 2 waters. It doesn't. It's neither here nor there. However, if you start off with FADH2, you don't even use complex 1 at all. Okay, so FADH2 will start off and it will pass its electrons to complex 2. Complex 2 passes it to to coenzyme Q, which passes it off to complex 3, which passes it off to cytochrome C, and complex 4. So if you notice, protons only get pumped across for complex 3 and for complex 4. But since you don't use complex 1 at all, you don't ever pump those protons across. Okay. So then what happens to all these protons? We keep putting more protons on the outside here. They have to go somewhere. What they do is, and it really does look like a turnstile. It kind of reminds me of, like if you try to go to Disney World or SeaWorld or something like that, and you got to go through the little turnstile. The protons kind of go through the turnstile, and as a function of that turnstile, and I'll show you, hopefully I can get the video to work. It literally forces ATP to be synthesized. Okay, so it's like turning the wheel, and so that's what happens. Okay. So at least anecdotally, or qualitatively, probably not anecdotally, you should be able to see why NADH will make more ATP than FADH2. Does anyone want to hypothesize why does NADH ultimately make more ATP than if you start off with FADH2? It pumps more protons across the membrane. See, this shows 6. Like I say, some of them will say as many as 12. That's neither here nor there. But it pumps between complex 1, 3, and 4. It pumps that many protons across. So there's more protons going to go through the turnstile. If you start with FADH2, complex 2 does not completely transverse the membrane. And there are no protons that get pumped across in complex 2. So you only get the protons from complexes 3 and 4. And so that's why you can get up to 2.5 moles of ATP made for every 1 mole of NADH, but you only get up to 1.5 moles of ATP made for every 1 mole of FADH2. All right. Yes?
Why is it that FADH skips complex 1?
We'll get to that. Yeah, there is a very, very specific reason. Okay, so then, knowing what's happening here, and it doesn't matter where we start. I'm going to actually start with the intermembrane space. If protons keep getting pumped out to here, is the concentration of the hydrogens out there going to be high or low? The concentration of hydrogens. It's high, so is the pH going to be high or low? Low. You have a low pH here, high concentration of protons. So then what's the concentration of protons here in the matrix? Is it high or low? Low, and so the pH would be high. And so that's why it depends on, be careful of the wording. And see, people get it mixed up. You always have to remember that it's an inverse relationship between concentration and P whatever. And so if the question's asking for the concentration, remember it's going to be higher concentration of the protons in the IMS But if they ask you with respect to pH, it's going to be a lower pH. Yes?
Does like amino acids, you know how like if it's a high concentrate or a high pH, it pulls off their protons? Remember?
Yeah.
Is that NADH and FADH like that too? Do they have?
I mean, anything that has a titratable acidic hydrogen can be affected by the pH or pK, and due to the pKa. What we see here with the NADH and FADH2, it's not just the hydrogens that's being pulled off, it's the hydrogens with electrons. So it's a little different. It's not an acidic hydrogen in that sense. But you're right, if it was like, if we're talking about an acid, then yeah, it would affect whether the protonation state.
Since the outer membrane is more permeable than the inner membrane, is the pH concentration and the inner membrane space more similar to the cytosol?
No.
Okay.
Okay, not if you're healthy. If you're healthy, no. Okay. And we'll actually talk about at the end of this chapter, like what happens whenever the membrane, so the outer membrane becomes fenestrated, starts to break down, becomes holy. Okay. So yeah, that does play a very important biological role, but not in this. Not if you're a healthy person, or if it's a healthy cell and healthy mitochondrion. Okay, so we actually answered all those questions right there. Are there any questions so far? Anything else? We're going to break this down. Trust me, they've got the crystal structures for a lot of these, or at least part of the crystal structures for a lot of these complexes, which are major feats. Okay. By the way, I worked on complex 3 for my second postdoc. So a little disclaimer. Here's just a different. I wanted to make my own. One of these days I'll make my own because all of these look like they're ancient. You know, it says 2003. But it just kind of shows you, it's a little different way of showing the same thing with respect to pH and concentration, in case you need to understand that. It need a different way of looking at it. Okay, so this is the electron transport chain. So what happens is by pumping all those protons out, you create this voltage gradient. That's why it's called the electrochemical potential. And because of that, all this is saying is that those protons have got to go somewhere. They're going to go through, I call it the turnstile. It's really ATP synthase. But it's going to go through ATP synthase. And it literally, mechanically forces the phosphorylation event of an ADP inorganic phosphate to make ATP in water. But, yeah, so, and I do expect you to know the different, like how that occurs. But we'll take the time to walk all the way through it, and hopefully if I can get the video to work, there's a cool video. There's no sound to it, but you can see what happens. Okay. So I'll give you a moment in case you wanted to write that down, because I just tell you the 4 complexes, like I say, the 4 complexes, they have multiple names. So I'll tell you what they are. Sometimes they're just referred to as the Roman numerals, complex 1, 2, 3, and 4, and they do use Roman numerals. Okay. I do not remember that song. Alrighty. Here they are. I don't offhand remember what electron transport. Oh, I do remember. Yeah, okay. Yeah, it won't work on this because this is an iPad, but on the original PowerPoint, if that video is still up, then it will work. Okay. So, like I mentioned, they have Roman numeral names, so they'll say complex 1, 2, 3, and 4. But then they have very specific names as well, which it looks really difficult, but they literally tell you what they do, okay, for the most part. But each of them has at least 2 names. So the first up is complex 1, which is also called NADH, Ubiquinone Oxidoreductase. So and I should have put a box around this, because then you could probably guesstimate. If you want to just take a moment to think about the name, NADH, Ubiquinone Oxidoreductase, what kind of reaction is it going to be doing? It's a redox reaction, okay? And it's going to be, that just means transferring electrons. And so what is it transferring electrons between? if its name is NADH ubiquinone oxidoreductase.
NADH.
NADH and ultimately ubiquinone, which is coenzyme Q, by the way. I don't know if you know that. Okay. Or it's one form of coenzyme Q, which we also abbreviate, in case you didn't know, as CoQ. 03, and we'll draw them out and I'll explain what they're. But coenzyme Q has 3 states: the fully oxidized, the fully reduced, and it's half. And we're going to go through and we're going to fill in all of this and draw it in, okay, for each one of these. But yes, NADH is what's going to give up its electrons. It's going to transfer electrons to a host. It's not just FMN, but a host of different other in-betweens. Like, what's a good word? Like arbitrator? That's not right. I can't think of a good word I'm looking for. But you know, like go-betweens between NADH and ultimately to coenzyme Q. Coenzyme Q starts off in the ubiquinone state that becomes ubiquinol, which will make more sense whenever you see it in this moment. Okay. Then complex 2 actually has 2 names. Well, 3, if you want to count complex 2 as being named. One of them is Succinate Dehydrogenase, and another one's called Succinate coenzyme Q Reductase. So what kind of reaction, if you just look at succinate dehydrogenase, what kind of reaction is it doing? Based off of its name. Based off of its name right here, what is this doing? The dehydrogenase, what type of reaction is it? It's a redox reaction, and it is transferring electrons from, it uses succinate. Where have we seen this before? This is the citric acid cycle. We've seen this is the actual enzyme in the citric acid cycle. So if you think back, and I said it, I don't know if you made a mental note or actually wrote it down. Remember I said that there's one enzyme of the citric acid cycle that's linked, and it's linked directly, by the way, to the electron transport chain and the succinate dehydrogenase. Succinate dehydrogenase is part of complex 2. It's also the only one, remember, that does not completely transverse the membrane of the complex 1, 3, or 4 at least. And of citric acid cycle, it's the one that makes FADH2. So it doesn't really have to leave. The FADH2 then just turns around and will pass off its electrons through various helpers, ultimately to coenzyme Q as well, to ubiquinone, which becomes ubiquinol. Okay. But that's the reason why, how did you word your question?
Oh,
About NADH and FADH2, but I don't remember how you
why
worded it.
does FADH2 skip?
Why does it skip? It's because the enzyme that it skips is in complex 2. So we got the FADH from citric acid cycle, and it's already part of complex 2, so it does not. There's not a binding site for FADH2 on complex 1. Just like there's not a binding site for NADH on complex 2. They're specific for their original electron donors. Okay. All right. So then we have complex 3, which is called complex 3. It's also called cytochrome BC1 reductase. Sometimes they also just call it the BC1 complex, which I may have the habit of calling it that, because those of us in the biz, that's what we used to always refer to. Really have to write all of that down. And I apologize, I do not, and historically I don't know why. If you notice, we've talked about cytochrome C, we have cytochrome BC1, we'll have some of these, we're going to talk about hemes. They have really weird names like heme A and heme A3. Don't ask me where heme A2 is. It's not there. So I don't know. I don't know how they came out. Why did they only use the letters A, B, and C? Why one of them is called C, one's called C1? I have no earthly idea. Okay. But yes. So cytochrome BC1 is also an oxidoreductase. So it does a redox reaction, but it's where the coenzyme Q in its ubiquinol state returns to ubiquinone state. It transfers electrons. It's really complicated. It uses something called the Q cycle to ultimately give it to cytochrome C. I like to use the word jump, because cytochrome C is the only protein in this entire process here that is not an integral protein. Remember, integral means stuck in the membrane. It's a peripheral protein, and it's in the intermembrane space. So it's the one that's in the intermembrane space, and it does physically leave when it gets reduced, and it ultimately goes to cytochrome. It goes to complex 4. So cytochrome C, which the jumper, okay, it's going to go to complex 4. Complex 4 is called cytochrome C oxidase because it oxidizes cytochrome C. It's pretty simple. And the electrons get transferred via hemes and coppers ultimately to oxygen to make water. Okay. Like I said, we are going to break these down individually and go over all of them in great detail. The first thing that I wanted to address, though, is that coenzyme Q, or CoQ. Okay, I'll give everybody a moment. I'll pause it. This is coenzyme Q. I will not have you draw it. I would, especially, this is the isoprenoid. Do you remember the isoprenoids from last semester in chapter 10, I think it is, when we talk about lipids? And it's a type of building block for lipids, and they are these 5 carbons that go like single double bonds, and they can be made. Well, coenzyme Q is made from it has an isoprenoid tail, like farnesyl groups and geranyl groups and all that kind of stuff. So it does have that. What's the whole purpose of this tail? Does anyone know? If you look at the chemical characteristics of all these carbons and hydrogens, what should stand out? What's the overwhelming chemical characteristic of this? It is extremely hydrophobic, so it never, ever leaves the membrane if you're healthy. It's stuck in the inner membrane of the mitochondria. Coenzyme Q is, at least for the Electron Transport Chain. Don't ask me how I got there. I don't know what inserted it there. But this is the business end. Okay. As I mentioned, it has what's called a ubiquinone. See, because it's got this diketone. And that's why it looks a lot like the quinonoids that we talked about last chapter. It's got a ubiquinone, and on the other end, the far end, it's got the ubiquinol, because now it's got 2 alcohols. It's a diol. Okay, so because of that, it can actually take up to 2 electrons and 2 protons, and it has a free radical intermediate that is relatively stable. If you think back to organic chemistry, you may remember that these radicals here, because of resonance, are more stable than what you'd expect. And so since it's halfway between ubiquinone and ubiquinol, it's called semiquinone. And they also abbreviate these as Q, QH2, and then the QH with the radical. Okay. So, knowing what you know, based off of organic chemistry, is ubiquinone fully oxidized or fully reduced? It's oxidized. See, we got lots of carbon-oxygen bonds, so this one, therefore, is going to be fully reduced. Okay, this is half. And so, yeah, that's why that. Okay, and so, whoops, that's what this is just showing you right here. But once again, if I say ubiquinone, I'll try to always be precise, ubiquinone versus ubiquinol. And if I want to speak in broad generalities, I'll try to remember to say coenzyme Q. I'll try.
You're saying ubiquinone can be CoQ?
Yeah.
Okay.
Well, I mean, coenzyme Q is all of it. It's got 3 states.
But more likely it's being referred to the first one.
That's why I will just always try to be precise and say ubiquinone, or write out Q. Like if it just writes out Q, that means it's fully oxidized. If we write out QH2, it's fully reduced, and it's ubiquinol. And well, the semiquinone is semi, so it's half. So that was the easier one to, and it's QH radical, or radical QH. That's why. And you'd actually, I don't I don't know, you probably had to learn the electron mechanism, like the electron pushing mechanism from organic chemistry, or something like this. I used to do it. Typically, it's probably in the phenol chapter if you wanted to go back and look at that. Because, this is the type of phenol, and so it's just a special property for the aromatic ring. Another, to review, something that we've already talked about once before, is iron sulfur centers, or these iron sulfur proteins. You can ignore that question because I've got the answer there. Part of the enzyme can be the iron sulfur center. It's cysteine. It's the answer's on here. But we've already talked about this. We talked about, remember what enzyme we've already talked about that had an iron sulfur center in it? Is the conatase, had the Ferris wheel. And so these are some of the different geometries that these iron sulfur centers can have. And I'm not an electrochemist, but based off the geometry, it affects its electron potential and its ability to transfer electrons. And so that's why the geometry is important. But don't ask me how or by what much, which one is better in this instance or that instance. Okay. This is out of order. Because we're going to see, and a lot of times they're just abbreviated as FES. Sometimes they get really specific, and they'll say like Fe4S4, 1, 2, 3, 4, 5, 6, or something like that, to where it lets you know how many irons and sulfurs are total. But usually, we just call them. They'll just have it as FES, for an iron sulfur center, iron sulfur protein. All right. I liked this picture down below, the one on the left. It just looks really cool. The little electron gradient thing that's showing here. But this is just a repeat of what's going on. The one thing I don't like, is this one? No, never mind. This, I took out the one that, I must have taken out the one that had numbered incorrectly. But this just helps to show like what's going on as well. Okay? So I should move that one up. But they couple the two reactions together. So we have the electron transport chain here, then we have oxidative phosphorylation happening at ATP synthase. And please note, it's called ATP synthase, not ATP synthetase. Oh, right. This, I really, really like this figure. I'll give you a moment to look at it. This is the ATP synthase. And very similar to, remember how hemoglobin had what they call the alpha and beta subunits, but it didn't have anything to do with alpha helices versus beta sheets? Something like that is happening here. Just because it's alpha and beta, it's not because one's alpha helical and one's beta sheets. Okay, so if you notice, it's not labeled on this side, on this graph or picture. Which side is the matrix, mitochondrial matrix, and which side is the inner membrane space? Is this one down here at the bottom, is this the IMS, or is this the mitochondrial matrix? It's the IMS. This one's the mitochondrial matrix. How did you know that? Concentration of the protons. Okay. I like this one because of the fact that I've given this figure before and people have labeled it wrong because they're just used to seeing the orientation where the IMS is on the top versus the bottom. But remember, it's in 3 dimensions. You could even do it on the left or the right or anything like that. And so, right. As I mentioned, this really does kind of look like a turnstile. This is called the stator protein. It's like a little ratchet that helps hold it in place, as it goes back and forth, back and forth. And so there's the alpha, beta, the gammas, subunits. They call this part F1, this part F0. And so sometimes it's even called an F1, F0 ATP synthase. And it's based off of, I think, the bacterial naming system. That's why that says those two. And the reason why is because F0 is the part that's hydrophobic and stuck in the membrane. F1 portion is aqueous and stuck into the mitochondrial matrix. So it kind of looks like a little mushroom sometimes whenever it gets drawn. Okay. What happens is the protons will actually go through the turnstile, and it literally does turn. I mean, it literally turns this, and I'll show where the ADP and then inorganic phosphate will bind into the region here, and then as it gets turned, it gets forced to be made into ATP, and then it gets released. Okay. The term that they like to use for this is called chemiosmotic coupling. Just an FYI. I probably won't say that a lot in case you're reading it, or if it's on one of the questions or something. It's just a fancy term. Okay. Of everything that we've been saying so far. This is the fancy way of saying you made that electron voltage gradient. Okay. Are there any questions so far? I said all this is literally just another way of wording the same thing. Are there any questions?
So you're saying this is just one word that explains what you just said?
Right. I mean, as I've been explaining over and over again, we've got protons being pumped across into one location as electrons are going transverse, according to that, or orthogonal with respect to that. It's not, they like to say it's asymmetrical. They're not symmetric. Like the proteins are not symmetrically there. They only go one direction, okay, in order to create this this proton potential gradient. And so then what happens is the ATP synthase will take those protons, shuffle them through the turnstile, for lack of a better word, and that's what will ultimately make ATP. So we're coupling those two pathways together in this instance. Okay. I really like this little course. It doesn't show complex 2, but it does show what happens from NADH. Okay, once again, you could even take this and draw your own version here, starting with complex 2, because from this point on, it would be the same. Notice it does not show ATP synthase. It's only electron transport chain. This is the kind of thing that we're going to be discussing in great detail. So there's the intermembrane space, there's matrix. It's showing you the flow of the electrons, what that means. Of course, you already understand that, from organic chemistry. But the NADH becomes NAD+. There's FMN. There are these iron sulfur proteins. And in the process, FC, if you notice here, it says 4 hydrogens. Some books say 2. I'm not so worried about the stoichiometry there. Coenzyme Q, what I don't like is they don't show the different versions of it going from ubiquinone to ubiquinol. Okay, and then back. This is the Q cycle. This is definitely not, I don't want to say dumbed down, but simplistic. There's a lot more that's going on. But then we have complex 3. And what's cool here in what happens with the Q cycle is only one electron goes down, for lack of a better word, and one electron goes this pathway. So because the reason why is coenzyme Q, remember, it accepts 2 electrons. But cytochrome C can only accept 1 electron at a time. And by having the Q cycle occur here, you don't ever short circuit your machine. If you want to think of the mitochondria as a machine, it never short circuit. Well, if you're healthy, it won't short circuit it. Cytochrome C gains the 1 electron, jumps over to complex 4 where it passes it off. And once again, I told you that they have the really weird, don't ask me, I don't know where A2 is, but it goes from A to A3. There's also coppers that's not shown in here. And once again, more protons are pumped across, and then oxygen gets those electrons to make water.
FAD might react with CoQ before it enters complex. It's in complex 2.
Right, this one's not shown. That's why I say on your own, if you want to just have this as an example, you can even draw in, okay, this is complex 2 would be here. you ignore all this. Complex 2 is here, there's no protons that go across. Okay, but the electrons would go from FADH to FAD. The electrons would then be passed off to make ubiquinol, QH2, which then go through the Q
So
cycle.
they're both making the same thing.
And so, right, this portion on is identical whether you start with NADH or FADH2. NADH is the only one that goes through complex 1. That's why you get more protons. That's how you ultimately can make more ATP. But not every enzyme utilizes FAD and FADH2. I mean, NAD, NADH. Some of them utilize FAD or FADH2. This is why we do have another way to also make to use that for our advantage to making, ATP ultimately. Okay, I think this is an awesome place to stop because I'll just show you a little taste of what's happening next. When I say we're going to go into detail, we're going to go into detail. Okay, so that's why we will break it down. This one, by the way, you can either think of it looking like a gun or looking like the state of Florida. I prefer to be on positive and think state of Florida if you turn it the other direction. So, okay, but that's just a little foretaste. That's what I mean by we're going to break it down in further detail. Go ahead and put all of it in. It kind of looks like the letter F, kind of looks like a case of the direction Florida, a gun, gamma, whatever, floats your boat. Okay, this is complex 1, right? So complex 1 so far, they don't have and I haven't checked this year, but they don't have the entire thing crystallized yet, but they do have this portion that's the aqueous portion crystallized where you can see the actual electron flow up through, since I'm comparing this to Florida, this would be through peninsular Florida versus the panhandle. Okay, so it's easier for me to refer to it as that. All right, so the panhandle would be the portion that would be, stuck in the membrane, and the peninsula would be what's in the mitochondrial matrix. One other point I want to make out, whenever they're talking about the mitochondria, I don't think I already discussed this, but another nomenclature that they like to use is called stuff N side, and it's not because it's on the inside of the mitochondria, it's in the matrix, but the matrix is the N side because it's negative. And then the P side is the intramembrane space because it's more positive because of the influx of protons. So sometimes whenever you discuss these complexes, they'll talk about the P side versus the N side. Just like in anatomy, we talk about dorsal and ventral and that kind of stuff, or posterior and inferior. So that's another way to do orientation. It's not that the protein itself has a positive or a negative side. It's talking about with respect to the mitochondria. All right. So what happens here is the NADH is going to donate its electrons to form NAD. Okay? These electrons get passed to a flavomononucleotide, an FMN flavoprotein. There are multiple proteins. So for those that, this would be an example of quaternary structure. There are multiple proteins throughout this entire process. So the FMN accepts the 2 electrons, and then it passes it kind of like, it reminds me of a ping pong machine, because it's going to pass the electrons through a whole series, not just 1 or 2, but a whole series of these FES centers. One to another, to another, to another, to another, okay? Until it reaches I'm been calling the panhandle, the part that's in the membrane arms, it's shown here, okay? One of the subunits here is called the N2 subunit, and I don't know why, okay? It is also an iron-sulfur protein, but this N2 subunit is what's going to hand off the electrons ultimately to coenzyme Q. So this would be the quinone. This is the quinol form of coenzyme Q. Okay? Now, they don't know the exact mechanism with which it happens here. Okay? And if you notice, 2 protons go through here to make the QH2. Up to 4 protons. Like I said, there may be some books that will tell you that it's only 2. But up to 4 protons goes through the panhandle region as well, then transverses out into the intermembrane space. There is a putative Q cycle of some sort that happens here, but they have not. There's not they've not been able to prove that. Okay, it's a putative based off of complex 3, which we'll get to in a little bit. They believe that something similar to that is happening in complex 1. Okay. And so that's why on your landscape portion of the membrane, you can start to draw in some of this stuff. So for example, and I probably won't. All right, are there any questions over complex 1?
The way you drew it looked like the QHC, what's that called?
That's the ubiquinol,
Yeah,
the reduced CoQ.
Does it stay in the membrane to go on to complex?
Yeah, 3.
Okay,
Mhm.
so it never goes back.
It will not, it never leaves. It stays within the electron transport chain. It will never venture into the mitochondrial matrix or the intermembrane
also
space.
still in.
Mhm.
Okay.
Yeah, the entire Q cycle is within either one of the complexes or the membrane, and they do not venture outside. Okay. For every single one, you know, you could draw in, and it doesn't matter. I'll use a different color just to show you. NADH plus a proton goes to form NAD. Okay, and so then the electrons, so this will be electron. I'll do electron flow, you know, in a different color to show you. You know, go ultimately to FMN, not ultimately, I'm sorry, first to FMN, and then it goes through, they go through a series of FES proteins to ultimately get to the N2 protein. The N2 protein then passes the 2 electrons off to coenzyme Q. Oops, wrong one. Whoops, I've already run out of room. And the coenzyme Q in the ubiquinol is what will go on out into the membrane, okay? And at the same time, you've got, you know, 4 protons going through. Once again, this is all still the plasma membrane. Not the plasma membrane, I'm sorry, the mitochondrial intermembrane. Okay, so that's to give you an idea of what I was talking about. Just like, I forgot to put how Q got in there. Whoops, that's supposed to be a straight line. I'm really having problems drawing. The Q did not leave the membrane, contrary to what it kind of looks like. Okay. All right, so that's how NADH got started. Now this is FADH2 for complex 2, which once again was Succinate Dehydrogenase from the citric acid cycle. It's just that this is the portion that would be from the citric acid cycle, okay? But it's all part of this huge complex. And this was the portion of the 2 pathways that cross over, okay? This is where we have to keep some of the certain things in mind. I'm going to go ahead and walk you through all of this and try to point out some of the important aspects. Remember, this has nothing to do with complex 1. It doesn't, yes, it's got that one little alpha helix that kind of like sticks up like a little periscope, but it does not really go all the way through the membrane to where the active portion of the enzyme doesn't go all the way through the membrane. Just put it that way. And it is true that it's also really, really huge. They're showing it with, you know, the how lipids attach and all that kind of stuff. Well, okay. All right. So some of the things to keep in mind here are whether or not protons pass through the membrane, because they're not drawn in this instance, and the fact that we're talking about how in the citric acid cycle FAD became FADH2, then the FADH2 goes back to FAD by passing off its electrons. And in particular, I want to point out this heme B, that's this thing right here. It plays a very important role, but it's not. If you notice, the electrons, which what is that color? Is that blue or green? Blue, I think. The electrons normally don't go there. So what's its role? It's not there just for looks, you know, what's its role? And so I want you to think about that in just a moment as we go through this. Okay, so as you draw it, we have citric acid cycle. We had FAD becoming FADH2. This is the crystal structure, so they're only showing it with FAD because if you had FADH2, the reaction would occur, right? So this is with it, but you can see where the binding site is. Okay, so the electrons first get passed from the FADH2 through a series, once again, of these different types of iron-sulfur centers to the waiting ubiquinone. Remember the none? That's the Q, whenever they just abbreviate it as Q. And it becomes ubiquinol, which is QH2, and it leaves to go to complex 3. Remember, it has nothing to do with complex 1. Complex 1 and complex 2 are separate from each other. As I pointed out, we didn't have anything. There's nothing to do with heme B. So what is the role of heme B? What do you suppose is there? It's not in the normal flow of electrons. What do hemes do? Pardon?
Metals.
It's got a metal. Usually what metal is it? Iron. Okay. So what can that do? What can iron do? One is combine oxygen. Yep. What else can they do? So that's like the hemoglobin. What's the oxidation state? What's the charge on the iron in heme? So you have like an iron 2 plus. What else could it be? Like iron 3 plus. So in order for go from 3 plus to 2 plus or 2 plus back to 3 plus, what must also be happening? If it's changing its charge, it's electrons. So you can imagine this thing is having electrons are transferring, because I can't speak that fast. The electrons are transferring so fast. I don't know the exact numbers here, but I know from working on complex 3, we're talking for a normal healthy individual, it needs to happen 100,000 to 150,000 times per second. They're flying. This is where the rule of thumb that you probably learned in organic chemistry can be violated. Remember, maybe, I don't know about your class, if I ever told you this, but a lot of times whenever we're doing electron pushing, we would say, electrons don't just pop off. Like you would have those flying electrons, like sometimes I'd have a student I'd take off a half point, where they just look like little eyeballs, like floating around. They didn't. They would go and attack things, but they're not just going to float around. Well, here, the electrons are having to fly so fast that occasionally, they can fly off. So what would happen here, let me put it so that way it comes on the video. What would happen here is, as they're flying through here, if the ubiquinone's not there and ready, or not in the right orientation, it could fly up, okay? So you want something, and this is my word for it, like a sponge or like a catcher's mitt, if you think about baseball, to catch that electron and toss it back in. Toss it back in if it gets by. And so in doing that, it's going to keep the electrons, because just what's true from organic chemistry is you're not just going to have an electron like floating around out here. It's going to go and attack something, right? And what can attack is it could reform reactive oxygen species, which would be bad. Okay, so it helps keep those free radicals from forming. Okay, so it catches it, tosses it back, catches it, tosses it back whenever the Q is ready to become QH2. And so it's like a safety valve, if you want to think of it like that. Okay. And so once again, we've made QH2. Ultimately, QH2, once again, it stays here. It's going to travel onto complex 3. Just like the QH2 that was made from complex 1 can travel onto complex 3. It never, ever, ever, ever goes from complex 1 to 2 to 3 to 4. Okay? It's always going to go 1, 3, 4. I'm sorry, technically 1, 3, cytochrome C4, or 2, 3, cytochrome C4.
What's ROS?
Reactive oxygen species. Those are bad, like superoxide, hydroxyl radical. Those are the two biggies. Hydrogen peroxide technically is. You can also have them, because then they'll go on, they'll do, what do reactive oxygen species? You have to have some. We have enzymes that take care of it, like superoxide dismutase, that's what it does. Peroxidase is what it does. So, but what happens if you have ROS forming? What does it go and attack? What are some of the things that these reactive oxygen species can attack?
Doesn't it break down?
What?
Doesn't it like break down?
I think what does it break down? What can it break down? It can break down DNA. It can, well, there's no DNA there. So what else can it break down? It can break down proteins. And what else can it break down? What else is here in this instance?
The membrane.
The membrane. It breaks down anything. I mean,
So
it
that's
can
why
break
you
down.
want antioxidants.
Yeah, your antioxidants break down, are there to attack reactive oxygen species, like vitamin C. Okay. Any other questions on complex 2, succinate dehydrogenase? Because I really want to spend some time on complex 3 because it's, first of all, I worked on it, and secondly, these are relatively simple. Complex 3 is hard to even draw. If you thought these two were big, complex 3 is massive. I don't remember now the exact number. I want to say 20 some odd subunits, like different peptides and proteins all come together. Because technically it's a dimer of polymers. So each complex 3 has multiple proteins, and 2 complex 3s come together kind of like, what is that, the famous sculpture, The Lovers, or whatever, where they are intertwined. Okay, so that's why if you notice it looks like everything is symmetrical, it really is. So they're huge. It's called the BC1 complex, and that's because, and once again, I apologize, I don't know why, I don't know why I should be apologizing. They really stick with the letters. They don't give, if you notice, see we got a heme BL, we have a heme BH, we have cytochrome B, we've got cytochrome C1, we have heme C1, and then we have cytochrome C, which isn't even pictured here, but that's what, that little dash. So I don't know why. I have no idea. Why couldn't they just say, you know, like A, B, C, D, E, F, G, H, you know, that kind of thing. But they didn't. Oh, I forgot the answer to the question, I've addressed it multiple times, but no protons pass through, once again, in the membrane in complex 2. Protons do pass through the membrane here in complex 3. All right. So, this is complicated. All right. I'm going to walk you through it once, and then, let me make sure that the next figure come in. Yeah. Then we'll walk through it at least another time or two. Because it will probably, it's kind of hoping you try to read, especially what is that figure in the old book, it's figure 1912, so it's probably about the same figure in the new book. I worked on this complex, and it took me multiple times to try to figure out what they were trying to show. Okay, so remember we had QH2. So the QH2 comes in and it binds. Now the problem, and ultimately cytochrome C, and it does have, you know, it's got its cytochrome there. Cytochrome C is a protein that's going to get the electrons, but it can only take 1 electron at a time, and QH2 has 2 electrons to give. So when QH2 comes in, it's actually going to give off the electrons 1 at a time. Because coenzyme Q, one thing that makes it really special is yes, it can, most things can, you know, maybe it can accept 2 electrons, but then they'll give away 2 electrons. This one can actually, coenzyme Q can form what's called the semiquinone, which is actually a radical. And it's semi-stable, more stable than what you'd expect. So what it does is whenever it binds, it's going to pass 1 electron up towards the P site, towards the intermembrane space. And it does so through this weird looking thing that kind of, to me, right here, kind of looks like a duck head. But it reminds me a lot of, has anyone ever looked inside like a grand piano or a baby grand piano? And you've seen like the hammer, no, you know, it comes and strikes. That's kind of what they call it they call it the risky, R-I-E-S-K-E, it's called risky, the risky protein, or the risky iron sulfur protein. And what it does is it comes down really fast and it grabs 1 electron from that QH2 and flips it up towards, what am I thinking? Flips that 1 electron up using its iron sulfur center to heme C1, that's in the cytochrome C1 subunit. Then that electron can go on to cytochrome C to reduce it, and cytochrome C will jump. Remember, it's the only protein that's not in the membrane at all. It's going to jump and go to complex 4. but we left one of the electrons down. So this would then be a semiquinone, okay? That could be a problem. So what do we do with that semiquinone? And it's one electron. Well, what it does is it's one, oh, and by the way, one little side fact is the risky protein of one subunit actually grabs the electrons from the other subunit, not of the other cytochrome, the other complex 3. So like the levers, I'm not that jointed, but if this was the risky protein, it's actually grabbing the electron from this elbow to flip it up, you know, and this one would grab it from the neighboring elbow to flip it up, and that way they're constantly going at the same time. That's why it's got to go really, really, really fast. Like I said, 100,000 to 150,000 times per second. Whenever on the wild type ones, whenever we were working with them. Okay, so the other electron goes through this lower potential route, okay? And if you notice, see, it's got this thing that says QP and QN. These are different binding sites. Okay. It's called QP because it's on the P side of the complex, and the QN is on the ends. I hate saying ends, like negative. The side toward the mitochondrial matrix. Okay. The reason why I wanted to point that out is the second electron goes down, for lack of a better word, to heme BL, then it gets passed to heme BH. Okay, and then heme BH will pass it off to the QN position. Okay. And the reason why, but what has to be there is there's got to be another Q already there. So at any point in time, you're going to actually have 2 Qs on each side of the cytochrome BC1 complex, of the complex 3. So the first Q grabs that electron and it becomes a semiquinone and it stays there. Okay. Because then what happens is another QH2 comes in. Oh, and that first QH2 was now a Q. It just goes out the back side. Another QH2 comes through. It puts one electron up here to cyclone C, and it's a semiquinone. And it's going to pass down the second electron to the previously semiquinone there, which becomes a QH2, because now it's got both electrons. And it passes, and it goes back up through, and it can do this entire cycle again. Okay. So like I mentioned, the Q cycle is not the easiest thing. This is probably I thought it was one of the more confusing things. And I really hate this figure, but I also don't know a better way to show it either. For simplicity's sake, they took away the handle of the hammer-looking protein and just showed its head. That's the head of the risky protein. So these are like I said, it took me forever. I'm like, what is this trying to show? This is the first reaction, the first time the Q cycle occurs. This would be for like the second time the Q cycle occurs. So when you add them up, this would be the overall reaction. Okay, so you actually end up, we will have 3 to 4 Qs for one complete cycle, because you have to do it twice, essentially. Okay, so I'm going to walk through it again. Once again, remember this is called the P side, the intermembrane space side. This is the N side, or the matrix side. So we actually have 2 binding sites that can have a Q on them. So we have a QHC that came from either complex 1 or complex 2. Right now we're going to just say it's complex 1 or complex 2. It can come from one other place as well. Okay. It comes in and it binds on the P side. It passes off 1 electron to the risky protein. Once again, the arm is gone, so that way you can see this binding site. It passes off 1 electron. It flips it up to heme C1, which flips up to cyclone C. Cyclone C leaves. Okay. And so it would look like a semiquinone. So it can't stay there. It's going to give up that second electron. The second electron goes this direction. Okay? And so it goes down to heme BL, which has the lower electron potential, to the heme BH, which has the higher electron potential, which passes it off to a Q that's waiting there for it. That Q then becomes semiquinone. See, it becomes the radical. That's what they're trying to show you. Okay, originally, this first QH2, now that it's passed off 2 electrons, it becomes just a regular Q and it can leave. And in the meantime, two protons have gone through. Now, this semiquinone can then shift over. That's what they're trying to show. That's what this is here. Now, that's where that semiquinone came from. That semiquinone was formed from the very first time. Okay, but it's not finished. Otherwise, you don't want it to leave just to stay like this. So what happens is another QHT comes in. Once again, it passes one electron off, like normal, towards the risky protein, cytochrome C1 to cytochrome C. Cytochrome C can leave. It passes that second electron, so that way this one became a Q, and the Q can leave. That that second electron goes down to heme Bl, heme BH. But now that's that semiquinone from that first round. That semiquinone now can become QH2, and it grabs 2 more protons to become QH2. It's got that second electron, and that QH2 can leave. That QH2 can now go on to another complex 3 and do this entire Q cycle over again. Okay, so that's why at all points in time, there are 2 Qs per, technically there's 2 Qs per unit. Okay, so there's a lot going on here. It's just, walking yourself through it over and over and over again. Once again, they're showing, the way that they have it, the one on the left shows like what happens when the first QH2 binds. Remember, there's already a plain Q down below. Then, and that plain Q became semiquinone. This one, the one on the right, is showing what happens when the second QH2 comes and binds to form this QH2, and then cytochrome C is once again reduced another time. Yes.
So this is referring to the 2 different sides of it, or is this all happening on one side of it?
Well, it's all happening on both sides.
Okay.
That's huge. And so they're just showing you one side. Well, technically, they're showing you 1/2 of each side. I shouldn't say 1/2, because technically this risky protein belongs to the other, like the back one, but it's acting on what I'm calling the front, cytochrome BC1, and so we're only seeing it from like what would be the front side. But the handle portion of this is touching like what would be the back one, and it can also be doing this as well, and it would be using the risky protein from the front one, like this. Yeah.
In all these complexes, it just shows that protons are just kind of flowing to the p side. Where are they coming from?
Well, they're coming from the end. Is that the matrix?
Yeah. How are they they're just flowing through it just shows an arrow of 2 protons just go straight up, like nothing shuttling them or anything.
Right. They do that a lot. Of the 4 complexes, this is probably the easiest one for me to understand where they're coming from, because 2 of them at every other time, every second time, 2 of them come from the matrix to reform the QH2, right? Every time, 2 of them come from the QH2 to form to leave the Q, and they just leave to go on out here. And remember, we're water-based, and so many times it's probably a water that's going to be ultimately taking it to form H3O+, or it could be lots of different things. Yeah, in biochemistry, a lot of times we just write down H+, and just show it. Because one thing that I think sometimes gets lost in gen chem and organic chemistry is the fact that most of these molecules have titratable acidic hydrogens on them, so they're popping on and off all the time. Like an alcohol doesn't keep its hydrogen on it. You put it in alcohol with water, that hydrogen will pop on and off constantly. So the original hydrogen, like if you took it, look at it half a second later, the original hydrogen, it had maybe on water number 13 over here, whatever, because they're just popping on and off.
So you're just separating the electron from that hydrogen, and then it becomes a proton.
Right, and that proton will just dissociate, will just leave. It's acidic in that sense. That's why they're trying to show, and then once again, that's where they use this N and the P to show you which side it originated from, or which side to which it goes. Okay. And so this first 2 happens, and that's why some books will only show 2 hydrogens occurring here. Some books will show 4 hydrogens. It's not so important to me that you say what the molar ratio is overall. Okay. Any other questions? Like I said, this one is, and there are lots of diseases and stuff associated with these. Many times, sometimes they're called cardiomyopathies, and so you may have heard that term, or mitochondrial cardiomyopathy. There are lots of diseases. Also, there are common drugs and poisons and stuff. I have to be careful. I don't want to become liable for something, tell you guys and read about later on that one of you guys murdered somebody from it. But, for example, toadstools. You eat a toadstool, you're going to die. Well, one of the main things for the toadstool is that one, the stigmatine. I can't remember. I don't know how it's right. But what it does is it comes in here and it short circuits it. It binds here and it prevents the lower one, the semiquinone spot. It won't allow it to happen, and you die. You go into mitochondrial distress, and so electron transport short circuits, and you die.
But are there also disorders associated with the heme B from complex 2?
I don't know. I mean, I've worked with this one, so that's why I know more about this one. I wouldn't be surprised that all 4 complexes, and ATP synthase for that matter, sometimes will have various diseases and disorders associated with them. Many of these are probably result in spontaneous abortions if they were very egregious. Some of the ones with complex 3 that I know of are associated with those diseases, those cardiomyopathies that you hear about, like especially with athletes where they were young and they just all of a sudden have a heart attack and die. Like at the prime of their life, they didn't even realize they had any heart problems. And many times it's with this. They have a mutation where it affects the efficiency of the electron flow. So you have to be really careful because if you think about it, your heart constantly has to have lots and lots and lots of ATP because it can never rest. Yeah. If your fingers rest, your arms, that's one thing. Your heart can't ever do that. And so then if these mitochondrial cardiomyopathies start to build up, accumulate with issues with mitochondria, then you'll die. Okay. And now it's been several years ago, but when I first started teaching this course, it was in the news at that time. I forgot what. One of the Tampa Bay Rays, I think it was a catcher, and he was a really good player, and he had to retire really young because he was found out he was diagnosed with one of these cardiomyopathies. They said, okay, from now on, you're going to have to take it really easy. You can't do things that are really strenuous. So it's going to increase the likelihood of you having a catastrophic infarction of some sort. Okay, any other questions about this? Look, I had a great uncle, not a great uncle. He was a great man. I had an uncle that seemed to be in the prime of his life, and he was mowing. My grandfather had some type of dementia or Alzheimer's, and so he could no longer take care of the yard and stuff, and my grandmother was his primary care provider, so my uncle would go over and mow their yard and everything like that, and one day he was riding the lawnmower, and he just fell over, dead. He was dead by the time that he hit the ground. Another one, my aunt was there who was a licensed CPR instructor. They tried and tried and tried to get him, to revive him, but he never revived. And he was only 40, he's in his early 40s. And so most likely, it was due to mitochondrial cardiomyopathy. I don't want to scare anybody, so maybe I won't tell you. So, okay, I was going to say, because I mentioned, there's one hallmark of some of these diseases that can do like premature aging, because if you think about it, a lot of these can cause reactive oxygen species to accumulate, and so sometimes they can have hallmarks of premature aging. So if you see someone that's a little older than what they probably, it may be something like that. I don't want you to think that, oh my God, I got a gray hair and I'm 24 years old, so I'm going to die of a heart attack tomorrow. No, it's not. But okay. Are there any other questions? That's a bad thing when you start learning about medicine. I don't know, maybe it's just me. You start to come down with everything. No, it's like, oh, I can't find my fault, that kind of thing. All right. This one's complex 4. They didn't show cytochrome C. Cytochrome C, compared to everything else, cytochrome C is relatively mundane. It takes one electron, so it becomes reduced, and it jumps over to complex 4, and that's it. Okay, in this instance, that's it. Okay, but that's why it's not shown. But it would have come in and would have bound here. Okay. It doesn't look like there's, this one looks relatively small, and there are also mitochondrial defects associated with complex 4. Other people in my lab worked on complex 4. Okay. Now, up to 4 cytochrome Cs combine through the cycle here. Okay, I just wanted to point that out because, and when I say that, because of the fact that you're trying to make water ultimately. Okay, and remember, if it depends. If they show it just making 2 waters, then maybe it would only, no, I take that back. 2 waters would still have 4. 1 water would have 2. That, once again, the stoichiometry is not so important. But cytochrome C would be bound here, and it's going to be passing off 1 electron at a time. Okay, and so the portion, and I don't know about you, but I can't actually see what's going on here because I can't tell the difference in the colors. But there are coppers there, and there are also hemes, and hemes have ions. Okay, I can't see them. Okay. So that's why I don't want to show you this first, and then I'm going to show you the cheaper looking one that I can read. Okay. All right, so this one, I can read this one. I can't find it on the other stuff. So up to 4 electrons will ultimately be passed here. Cytochrome C, when it binds, is going to pass off the electrons through copper A. Okay, and copper A then will pass it off to the hemes, and it goes to heme A first, then heme A3. I don't know why there's not a heme A2, but there's not, okay? Sorry. And then it's going to pass it off to copper B. There's actually this, at least in our lab, we called it a channel where oxygen, if you remember, O2 is nonpolar, okay? Because there's not a dipole moment between they have the same electronegativity, right? It's 2 oxygens. So it can diffuse through the membrane. And so this channel would accept the oxygen, and oxygen's actually going to accept the electrons here as well as the protons to make water, okay, here within complex 4. At the same time, up to 4 protons, additional protons, are actually pumped from the N side to the P side as well. And once again, they in our lab we always called it a channel, but I don't remember how they got passed off. And there were people that were looking specifically, because it's actually fairly long distance, they were looking specifically at trying to figure out exactly how the electrons got passed from cytochrome C to copper. And I haven't kept in touch with them to know if they ever figured out what amino acids were involved in here or not. But they were looking at to see like what happens if you mutate specific amino acids, like how does that decrease or affect the electron potential and the ability for the electrons to flow. Because most of the other places, the ones that we looked at, they were actually really close together. This one's actually got quite a bit of distance. Okay. Are there any questions? That's why I said, oh, yours looks really nice. You know, to have like this as you go along, because it can help with following along and putting it all in place. Okay. Are there any questions with this? Anything with complexes 1, 2, 3, or 4? So what complexes will shuttle protons from the mitochondrial matrix to the intermembrane space? 1, 3, 4. So what one doesn't? 2. There have been times that I've just had quizzes or exams where I would just say, I would list something and I'd tell you, and I'd ask you to write down complex 1, 2, 3, 4, or the A for the ATP synthase, which we haven't covered yet. And I would just list stuff like that. Which ones, you know, and so you'd have to write 1, 3, 4. Which one is 2? Which one is associated with a citric acid cycle? 2. Which one has a Q cycle or a putative Q cycle? 3. Putative one was in. Where else is there a Q cycle? Possible Q cycle. It hasn't been proven yet. No, 3 has a real Q cycle because they've been able to do mutations on it and figure it out. 1. Okay. And so once you that kind of stuff where you kind of put things together. Which one has, you know, like the heme A's? This would be key. Now, if I just ask which one had hemes, then that's a little more tricky. You have to think there's heme B in complex 2, and at this row, we have heme A and heme E3 here. We have the cytochrome units that are affiliated with complex 3. Yeah. Okay, so, yeah, that's the kind of thing that could be asked.